These are the notes on Mendelian genetics. Just some basic terminology when it comes to genetics in the next unit, the Slice 9 genetics unit. If we look at, whoops, let me move this around here. And so you can see the nice picture there of Gregor Mendel, and he was the guy that was coined as the father of genetics. And he did a lot of work with pea plants, um, and he was a priest. And he lived in the 1800s, son of peasants, pretty poor, Czechoslovakia at the time, and he was just interested in how traits were inherited. He just had an idea, he just wondered, and that's a lot of what science is all about. People have questions and they decide to figure out or try and find an answer to their question. And so he actually worked with pea plants to try and figure out how traits were inherited. And so we started out with a what's called a monohybrid cross. Mono meaning a single hybrid cross or a single trait cross. And so he took two parents. He took again, these are pea plants. He took a smooth plant and a wrinkled plant, and he crossed them, and he came up with that most of the offspring were all smooth. And if we look at some of the vocabulary here, the P1, these are generations. P stand for parent. The P1 generation, again, the parents were smooth and wrinkled, and the F1 generation, F stands for filial or offspring, they were all smooth. And so that told him right off the bat that there was some sort of dominant law, that traits were dominant, and we call that as Mendel's law of dominance. If we The next thing that he did was he took all those smooth individuals in the F1 cross and he self-fertilized them. He self-fertilized them, and whoops, excuse me there. He came up with, this time, about 75% of them were smooth and 25% were wrinkled. That in that second cross that he did, he actually had some recessive traits. So he thought to himself, he did some more experiments, and he said that genes code for traits. For example, CJ. And he also said that, with each trait, there are alleles, different versions of the gene for each particular trait. For instance, smooth and wrinkled. And some alleles were dominant and some were recessive. And if the dominant allele were present, then that individual would have the dominant trait. So these were very important at the time. Again, we're back in the 1800s when he's talking about this, and it was very, very important data for scientists to go forward. He also concluded again that each organism contains two alleles for each trait. Again, you get, we talked about this in meiosis, how you get half your genetic information from the mother, half from the father, and in turn you have two alleles for each trait. And again, alleles are just the different versions of the gene. And so what he learned from this, and again, after studying meiosis, this kind of makes sense. He learned that the alleles separate during meiosis, that there are alleles on these chromosomes, and when the chromosomes separate, these alleles separate, and you get different again, combinations of the traits. And that separation of the alleles is called the law of segregation. So if we look at some more definitions, some more terminology that we'll be talking about throughout the course of this unit, we start with what we express alleles by, and that's just simply letters. If we have a capital A, then that is going to be represented with the dominant alleles. We always represent the dominant allele with a capital letter. Recessive alleles are always represented with a lowercase letter. And sometimes people tend to choose particular letters. For instance, the capital, whatever the dominant allele is, you should express, or if you're trying to figure out what letter to use, you generally use the capital letter of the dominant allele. So if we're using two letters such as red and white, red being the dominant, color and white being the recessive color, then we would probably use big R and little r, because red is the dominant trait, and so we'd use the capital R for that dominant letter. So again, we look at genes, are represented by 2 letters, and this is the next step. Now that we've figured out what's going to be dominant and what's going to be recessive, we have to also understand, again, back on the last slide, that each organism contains 2 alleles for each trait, that sometimes you're going to have 2 capital letters, sometimes you're going to have 2 lowercase letters, and sometimes you're going to have 1 of each. So if we look at what that particularly means, we have an individual that has 2 different alleles is said to be heterozygous. So if you were big A, little a, that's considered a heterozygous individual. Hetero meaning different. And that's also a synonym for a hybrid. A hybrid means different. If we look at any of the scenarios that would give you a big A, or little a individual, those would be considered homozygous. And a synonym for that would be purebred or true breeding. You might hear those terms come up when you're doing some Punnett squares. We also look at the combination of all the alleles an organism has is called the genotype. So these letter combinations, the actual letters, are the genotype. And that's written as big A, that would be that would be a homozygous dominant. Big A, little a would be a heterozygous individual, and a little a would be a homozygous recessive. Other definitions, if we want to take a look at the physical appearance of an organism, or what the organism looks like, that is the phenotype. Phenotype for physical. For instance, again, these are just words that we use to describe what the organism looks like. For example, an albino. When you look, and the reason why when you do the Punnett squares and we look at genetics, it's extremely important to take a look at both the genotypes and the phenotypes, because you can't always tell what the organism's genotype is based off the phenotype, because again, at least 2 letters make up that trait. So if an individual is dominant for a particular trait, you're not sure if it's big A or big A, little a. So you can't always tell that. So that's something to keep in mind when we're going through the Punnett squares, if you're not familiar with Punnett squares. The next thing, I've mentioned the word Punnett square before, and you all have done this before. If you've been in Fairfax County, you've worked with the Punnett squares. These are those little boxes that kind of look like tic-tac-toe boxes. And the reason why they're so important in genetics is that these little devices determine the likelihood of what the offspring could have. We're not talking about just the number of offspring that an individual can have. We're talking about the likelihood that those offspring would possess a particular trait. And they're very simple to do, and doctors use them all the time. You can sit there in the waiting room of a doctor's office and figure out particular traits, especially when it comes to whether the likelihood that your child is going to have particular traits. So if we look at the process, the first step is to draw a box, just like this, 4 squares. And the reason we have 4 squares is because we need to determine, of course, the whole purpose of this is to determine the genotypes of both parents and write them in the above box. So for example, if we have a big T, little t individual and a little t individual, and those 2 want to have offspring, we would use a 4 square box to determine what their offspring would look like. The letters on the outside of the boxes would represent the gametes. For instance, if this individual is going to donate, it can either donate a big T, because again, every parent donates half of their genetic information. So it can either donate a big T or it can donate a little t, depending on the chance that it's going to donate a big T or little t. The other individual, if you want to split that in half, the individual can either have a little t or a little t. Let's say that this individual is male So it's going to be donating sperm, and this individual is female, so it has the egg. So the female, her eggs are either going to be this little t or have this little t in it. The male is either going to have big T sperm or little t sperm. It just depends on which fertilizes the egg. Just give us a second. So when we look at the letters inside the box, what they represent, those are the genotypes. And so we have to figure out, when you're putting these letters together, we have to determine what the possible zygotes are. So you can fill in the box here. If this T and this T, if this is the sperm and this is the egg, if this sperm meets that egg, then the individual is big T, little t. If this sperm and this egg meet, the individual would be little t, little t. If this sperm if this sperm and this egg meet, then it's a big T, big T. And if this sperm and this egg meet, then it's a little t, little t. So you fill in the box. So that's not saying that we're going to have 4 offspring. That's not what these Punnett squares do. It just tells us what the offspring could potentially look like. So once we look at the possible offspring, there's 2 comparisons that we look at, the genotypic ratio and the phenotypic ratio. The genotypic ratio are the different letter combinations that you could get. So in this instance, you would have a possibility of 2 individuals that could be big T, little t, and a possibility of 2 individuals that are little t, little t. So that's about a 50-50 chance. If these individuals were going to have offspring, then it's about a 50-50 chance that or there's about a 50% chance the individual would be dominant, and about a 50% chance the offspring would be recessive. So if we look at the phenotypic ratio, what the physical characteristics would look like, again, if big T is tall and little t is short, 2 of the individuals would be tall and 2 of them would be short. So 2 comparisons that we look at when we look at these monohybrid crosses. So if we want to take a look at some human genetics, and again, we'll do lots of practice on the Punnett squares. If this isn't making much sense, then we'll come back and take a look. But if you just kind of want to get a glimpse of what we're looking at here, just some basic human traits. Tongue rolling is a trait, is an inherited characteristic. Widow's peak. Attached or free earlobes, being able to taste PTC paper, hitchhiker's thumb, bent little finger, mid-digital hair, dimples is a genetic trait, short big toe, short index finger. And so in class we'll come back and we'll actually do take a look at ourselves and do sort of a class sample of who's dominant and who's recessive. So this concludes the basic definition notes. We will come back to this presentation later on in the unit and talk about things like dihybrid crosses, where we're crossing more than one trait at a time. We'll look at more human genetic traits and some of the human genetic diseases that happen to occur. We'll take a look at sex-linked genes and instances that we have exceptions to these Mendel rules, where we have things called incomplete dominance and codominance. But make sure you have all the definitions in your notes and you are ready for class. If you have any questions, please feel free to ask Mr. Matthews.